Hi, everyone. I will be sharing my screen shortly. Thank you so much. In this session, I will be talking on molecular science and its contribution to health and well-being. This talk will lecture and give us a system biology perspective on the topic above. My name is Ayamfoluwa Alabetutu. I'm a molecular biology and genetics expert from Penza State University, Russia. I am a blood pathogen expert. I am also a bioinformatics mentor and community engagement manager with OmicsLogic Africa, Pan Biotech USA, and I'm one of the speakers of the Track 2 lecture series of the first International Symposium on Biotechnology and Molecular Science Development with the Faculty of Agriculture, University of Ilorin, Nigeria. I appreciate the Biotechnology and Molecular Science Committee and also putting this wonderful sessions together. It has been very, very interesting so far till this point. But the theme is very central and relevant to the current science advancement. And this being said, I would love to begin. I'd love to start by saying that molecular science, a multidisciplinary field encompassing the study of molecules, their properties, interactions, and behaviors today, plays a vital role in understanding the molecular mechanisms underlying disease pathogenesis in order to develop suitable diagnostic strategies and disease interventions. Many living cells phenomena, particularly those connected to diseases, cannot be explained in terms of single genes or just a couple of genes because of the strong interdependence between the molecular components of the biological system. So this lecture will showcase several multiomics cohort building tools, multiomics elixir in the clinic, genomics as a build-up from molecular scientific methodologies. So I will be following this content outline. I hope you follow us strictly. I will speak to the challenge and opportunities biology as a data science, the progress we've had so far, and a sample case. This being said, I would love to open by saying that it's precisely 19 years, 9 months, 16 days since the first human genome was sequenced on the 14th of April 2003. And now we live in a world of omics where it's all about genomics, proteomics, lipidomics, metabolomics, or let me also add the important omics, which is the economics of all of these omics. So it's very important that we think about how the omics revolution is interfacing with the genotype. We need to understand that a challenge, even from the beginning until now, still remain an opportunity for us to bridge the gap between genetic characteristics like phenotype and genotype. Still, we still need to understand the place of the multi-layer of molecular and other data types that we have and that molecular science has also generated. We need to understand that this discovery must be integrated into the health system. This being said, I need to speak to the fact that the incredible revolution we have seen over the past 20 years, the amazing health data that we have, it serves to enable us to look at all kind of diseases and health outcomes, and specifically about the molecular factors in between. And I will get to mention some of the many omics that have been measured in population studies, and building on these omics studies are yet ongoing to enhance our understanding of the causes of disease. Of course, this is actually one of the central themes for most research centers. I'm going to give us a flavor of the kind of research that is going on under research who that are interested in the molecules of electronic health records study, and there are some interesting aspects of the works in that areas which I'll take you through and give you one case study, including some related to genomics. If time permits, I will also touch on a COVID pandemic study of the first the first multiomics squad, or maybe some other multiomics squads. We speak or mention them as we go on. So how did omics get to this point? What is omics? What crosses your mind when you start hearing the word omics? I need us to understand that biology has evolved, and biology is rapidly growing to accommodate data science as a subfield. We learned that billions of data points from genes and other molecular analysis can be compiled into large files and systematically studied, which is to say that we have generated billions of data points, and that analyzing this data will actually lead to more knowledge and understanding about living organisms, including human, biotechnology, crops, livestock. Hence, the influx of data, associated methodologies, data-driven research makes system biologies or an aspect that I would love to introduce was called bioinformatics for life science as prominent as the emergence of computer in the tech industry. That being said, on the molecular factor preferences, we see various aspects of omics ranging from phenomics that is defined as the changes seen on an organism resulting in variation in the phenotype during the lifespan of the organisms. Genomics, which is the study of the whole genome of organisms, with the genome being a complete set of genetic information in an organism. Epigenetics, on the other hand, is a set of changes in an organism brought about by modification in gene expression rather than the alteration of the genetic code in the individual's DNA. Also, transcriptome, which is the total set of mRNA transcript obtained from the transcription of the genetic information and how it determines gene expression in a set of tissue at a respective time and condition. Also, I want us to understand that when data is being generated, those data are saved in a format, and this format includes the FASTQ, FASTA, GTF, and SAM and BAM format. The FASTQ provide us with reads from the sequencer. It saves files from the sequencer in a text-based format, and it stores the nucleotide sequence of the reads and quality score for each. Later, we have the FASTA, which is a text-based format for representing DNA and protein sequences. Typically, this is the reference file for the genome. Then we have the genome transfer format, which contains genome annotations. For example, the GTF will store start and end of a genomic element like the exons, the isoforms, and genes. So some files are the sequence alignment map that represent the alignment of these reads on the reference sequence genome. It usually starts with the header section followed by alignment information as tab separated lines for each read. The BAM file is like the SAM files, only just that the format is in a binary format. I need to also speak to the fact that the kind of data we have via NGS sequencing. Some persons may wonder what is NGS. NGS is next generation sequencing or sequenced data. So any data we get from our high throughput technology, Gs, from our sequence is called our NGS data. I would love to make a contrast between what we know as vitals and what is known as the molecular factors. Okay, So which of these factors are most important? There is going to be an interesting point I would love to raise here. So let me start or speak to the fact that medical data, or what we simply call the clinical data, is the type of information that has been used to define health and disease for centuries. So think of vitals. I mean, data points like temperature, heart rate, white blood cell count. This data is fairly small in size, easy to quantify, I know, intuitive to interpret. But especially once you get into the habit of seeing it, it is also surprisingly precise and useful when we need to detect a serious change in the condition and characterize it. But many broad categories of disease can be well characterized by these measurements that provide insight into the condition, etc. Okay. For example, what disease might this be, and where can you look further to treat a given condition? This clinical data is an indicator, but it's often appear too late. So many times when you get all of these informations, many times it doesn't serve the purpose of preventing disease and so on, and it does not provide enough details about the underlying causes and the mechanism of the disease. In other words, clinical data is just the tip of the iceberg that appears at the surface of what is going on. So you can just look at the screen and get to understand the reality. There are other types of data, molecular factored data that are interplaying in all of what we know as disease. With the advent of cellular and molecular biology, it has become clear that in many cases, changes in health, the emergence of diseases are driven by subcellular molecular mechanisms, and what appear to be a change in temperature started with a viral infection a long time ago before the temperature changed. So what protrudes as a lump, for example, or causes coughing and inflammation starts with a change in a single cell that lost control of proliferation. That's become clear that for many conditions, clinical data is more of a result rather than the reason. This understanding itself has since transformed biomedical research and the clinical use of high throughput data. And many ongoing studies are driven by the possibility of understanding mechanisms of disease and attempting to characterize disease at the molecular level using omics technology. When we talk about omics technology and biomedical data. High throughput biomedical data, we typically refer to the combination of clinical phenotypical data and the omics biological data, or the combination of observable medical phenotypes with information about underlying um, biological event. I want you to understand that for you to make the most of the multi-omics data, or maybe of the multi-omics era, there is a need for a biologist to become mathematically inclined. There is a need for the biology to understand that there is a place for machine learning, because machine learning can be used to process omics data sets and to find association with clinical data. All omics data high throughput. This means that the experiment themselves can be automated such that they produce a large quantity of observation for each sample. There are many omics, including phenomics. I think I talked about that information on the phenotype, how cell looks, the shape and types, while imaging is an important type of data that can be used, that can be processed using machine learning. But here we'll focus more on other technologies like NGS. So genomics, uh, for those ones that are joining us and are wondering what is genomics, it speaks to how that the DNA sequences that contains single nucleotide polymorphisms and other variations at the DNA level. So epigenomics speaks to the genetic variations, like histone modification, DNA methylation, transcriptomics gives us information on the gene expressions and its variation, like alternative splicing. Other types of omics data include lipidomics, proteomics, repeatomics, like you can see on the screen, and metabolomics. And some of these informations are actually acquired using spectroscopy. Other types of high throughput technologies exist as well. One example is one that comes in handy for drug screening, which involves an automated screening of molecules in cell culture. It's mainly used in pharmaceutical R&D to select molecules that have a specific functions. Hundreds of cells are grown in a culture, for example, and each treated with the molecules taken from a library of many thousands to detect the response of these molecules. Then they can then undergo automated imaging for a visual screen of morphological changes to the molecules. The possibilities are virtually endless in the multi-omics world, and all generated, and we generate a vast amount of data that would not have been possible to collect in some years back, especially when things were being done manually. Now, the process of uh, of this data transforming the quality of the details, transforming the quality of the detail into meaningful result for both biological and clinical standpoint is actually the reasons why we are doing this. Adoption of the next generation sequencing technologies, or the high throughput technologies, will allow researchers to understand many aspects of disease onset and progressions, and for the clinicians to be able to precisely diagnose disease and better understand the genetic risk factors for a disease, and to treat patients with the appropriate targeted medication. Real quick, I will just talk the fact that NGS allows researchers to be limited to explore genetic variations like never before, down to single nucleotide resolution. It's much more affordable now. Whether they want to identify specific variant from a focused set of region, or look more broadly for potential causative variation, and so on. So we have the whole genome sequencing possibility, where one can have a targeted view of a protein coding region of the genome, and so on. I am kind of trying to hasten things up. I just spoke a little to the whole genome sequencing, or the entire genome sequencing, and like that actually explains itself. There is an aspect of metagenomic sequencing that also involves the use of the 16S ribosomal RNA component of the ribosome, of the 30S small subunit of the prokaryotic ribosome. It binds to the Shine-Dalgano sequence and provide most of the SSU structures. Then we can also use amplicons in metagenomic sequencing for those that are interested. For those in microbiology and some of these other regions. So there is an application of high throughput sequencing here. I would love you to just go there, but this briefings speaks to a summary. It gives us a summary scheme of multiomics level with associated technologies, data type, shows all the different data types, the kind of output, the region, the type of research you can actually do with this type of technologies, the analytical considerations, research, clinical applications. It gives us 5 levels of components: the genome, epigenome, transcriptome, proteome, and metabolome, like what we can see, and all of which can be considered with respect to the phenome. Key associations between omics labels are also represented in this brief or in this scheme, including transcription. We can see an aspect of histone modification, TF binding, and actually I adopted this from a review of the application of high throughput sequencing in personalized medicine barrier and facilitator of future progress in research and clinical application paper. So I believe you can just go to the link and get to see this the more. I need to also speak to the fact that economics, that high throughput data or analysis of high throughput data is becoming affordable. So there's an economics of high throughput data in biomedicine. The growth in high throughput genomic data is largely driven by cheaper, faster, and more efficient sequencing these days. The cost of sequencing of sequenced genome in 2001, for example, like you can see on the screen, it cost 100 million, and in 2008 it dropped approximately towards 3 million, and today it's around $1,000. And this cost is kind of is just a sign to show us that the advancements in NGS technologies ever increasing the numbers of genome sequence, and the cost of sequencing has drastically reduced. So this is this is to support what Moore's law states, that we can expect the speed and the capability of a computer to increase every couple of years, and we'll pay less for them. That was what the prophet of that time says. So learning NGS-based technologies involves learning new computational methods and its transition application, as this is one of the key research area in today's world. NGS is starting to, provide powerful insight into genetic mutation, molecular network, and the underlying informatics of disease for a broad set of genes. As the cost of genomic sequencing rapidly approach the cost of many other standard medical tests, it becomes more and readily used both as a research technique. Here, we now move to the progress. What does the future hold? Since the uncovery of the human blueprint, we see how that many consortiums and reference genome targeted collaborations, samples, and databases have started springing up. We've seen multiplications of softwares to make studying of the omics data set easier. Tools and methodologies has been invented and has been worked on. At the same time, we see how that advocacy for bioinformatics training and education has been at the center of most of what we do. And at the same time, we see how that some cohorts has dedicated themselves for to explore health informatics, such that health records can and the omics technology know-how can be channeled towards precision medicine. That being said, I will just share some examples with us. We can see here how that the Human Genome Project was first introduced here by the chemistry, biology, physics, ethics, engineering, informatics. It started October 1, 1990, and was completed 14th of April 2013, and we were able for the first time to read the Nature's complete genetic blueprint for building the human being. Next, here we can find the E code, the gene code, where this consortium aims to identify functional elements in human genomes and model organism DNA, like the mouse, for example. From there, we can find the UK Biobank, which is the national flagship large-scale biomedical database and research resource for the UK. We can also find the international app map where we see collaborations of scientists, international scientists that seek to identify and catalog genetic similarities and differences in human being. They have collaboration between, collaboration and founding agencies from China, Japan, United Kingdom, Canada, Nigeria, United States, and so on. Then we can also see the 1000 genome research, which effort is to the 1000 genome projects. The aim is to create a catalog. They created a catalog of common human genetic variation using openly consented samples from people that declare themselves as LD. We have more. We have the UK 10K, which I would love us to check. Then the B1MG, you can just go there and go and check out so that you understand how the network of genetic and clinical data across Europe was kind of invented. From there, you can see the VGP, which stands for the Vertebrate Genomes Project, and we see how that they have reached out. Their aim is to generate an error-free reference genome of 70,000 plus genome extant vertebrate species. We see the Earth Biogenome Project that is aimed towards conserving biodiversity using genome sequencing technology, informatics, automation, artificial intelligence, and so on. We can see the African Biogenome Project, which is similar to these two. The aim is to deliver the value for the African people to build genomics bioinformatics capacity across Africa. We can see the 10P, the 10KP, 10,000 plant genome project, and they want to be able to represent 10,000 genomes of major clades of plant and eukaryotic microbes. Then we can see HDRUK, which is the Health Data Research Hub for the United Kingdom. That being said, I would go forward to talk about the sample cases that we have that I would love to bring here. I would love to make a case for integrative biology. I love to use a recent example to illustrate the paradigm here. And this is a work that has been jointly done between the University of Leicester and University of Cambridge using the UK Biobank International Flagship Population Cohort. I see how that these colleagues at the University of Leicester have spent several years measuring telomere length in all 500,000 people. For those who don't know what the telomere is, telomeres are protective ends of the chromosomes of DNA. As we age and reproduce and our cells reproduce, each time we lose a bit of DNA, so we have these repeat sequences of kind of nonsense DNA at the end of the chromosomes that don't have any genes in, and so that they don't have any genes, so they cannot protect the chromosomes. And these telomere lengths have therefore been of great interest in trying to and these scientists have tried to measure the telomere lengths to see is it a marker to how well or how badly we are aging, and what is relevance to disease. So we can see from this study the polygenic basics and biomedical consequence of telomere length abbreviation. We can see an integrative genetic studies, genomic studies merged or bridged with health informatics, health records, and we can see how that population of 500,000 people was actually the sample population. Now, so here we were able to link this telomere length measurement, of course, getting from the health record in UK Biobank. Looking at the observational association, you can see on the left between different telomere length and the different diseases. You can see, for example, there are positive associations with several kinds of cancers. For non-linear association with some diseases like leukemia, and even inverse relationship are seen with the liver cirrhosis. But of course, these observational associations may well be confounded. There may be consequences of disease processes rather than causes. So what we really want to get into is the understanding of the causes of the disease here. And here is some of the element of causality. And of course, the way they try to do this is by linking genetic data. So because we have the genetic data in UK Biobank across the whole genome, you can see here that you can then interrogate that that with a particular risk factor like telomere length, and in this case, and in this case, they found nearly 200 different regions of the genome that seems to influence an individual's telomere length. And of course, they try and they combine all of this together, bringing together the genetic data, the telomere length data, and the disease outcome data, try to understand which of this observational association might be causal again. And here is a screenshot. Here is a screenshot of some of the ones that is looking interesting. For example, you can see here that some of these cancers I showed you on the left side of the screen, those observational relation do appear to be causal, but there are association also in the opposite directions with things like coronary disease and the liver cirrhosis that we saw. So we can take one risk factor and look at the causal influence in a wide range of different diseases. And in fact, there is another paper in this regard that looked at the relationship of the telomere length in the UK Biobank with COVID-19, suggesting that that does seem to be that there is a relationship even after we account for chronological age and other potential confounders. So some further work here that has been done was that they try to understand the potential mechanism of association. Now, of course, as it's important to no longer think about just one risk factor now, but there have been evolutions in omics that allows us to measure many thousands of risk factors, and so rather than looking at just one genetic determinant and the causal consequences of one factor like just telomere length, we have seen researches that tries to examine or cross-examine many thousands of proteins. In conclusion, molecular science, systems biology, bioinformatics has gained root in Africa. Whilst the capacity for modern system biology research and training is available, but the burden of access to standardized laboratories in Africa and lack of financial prowess to get what is needed per time to employ bioinformatics techniques in research targeted at African data set still remain a challenge in biomedical science and agriculture. This development shows that when given the needed support, resource-limited African countries can contribute to the use of bioinformatics. It hoped that the identification of local institutions with expertise in system biology and bioinformatics will help to build partnership to develop peer support system among scientists and scientific organizations in Africa countries. Students, young and upcoming researchers may also explore some of the identified grants, research centers, ACE, consortiums, and note to benefit from scholarships, from knowledge, from fellowships, and so on. Bioinformatics of plants and animal research is still scanty in Africa and needs campaign to make it also prominent. I have compiled a list of short courses for you to be able to find free bioinformatics short courses here. And if you have any questions, please feel free to reach out to the symposium committee or reach out to me. Thank you so much for your time and have a great day.